In the marine realm, organic matter is produced almost exclusively where there's sunlight at the top. They're not limited by water, so this is very different than productivity on land, where water is one of the master variables. We have a lot of water in the ocean. You can quote me on that. That's a very, very almost always true. But we don't always have light, and the light is confined to the top. And I'm sure Brian has talked about this earlier in the week. We produce our organic matter, and then it sinks, and then some of it actually makes it into the sediments, but a lot of it gets recycled in the marine environment, which is of great interest for its own sake. But for me, as a geologist, I'm interested in that little tiny trickle of material that actually makes it out of this biologically active environment and gets preserved on geologic timescales. So it's just a little tiny sampling of what is up in the environment, makes it into the sediments. And it's transformed during that process of sort of moving out of the photic zone and into the sediments. This is a summary paper from Cindy Lee and Stu Wakeham and Carol Arnosti. It's a really great paper that pulls together compositional variation as particles sort of settle through the marine water column and make it to the sediment. That's the conveyor belt that brings organics into the sedimentary realm. And in this diagram, you can see the upper green bar here is amino acids, and the lipids are the black bars, and then the blue is uncharacterized. And if you also look, there's like a loss in the quantity. So we're like exponentially losing the amount of stuff. And the stuff that we're kind of getting at the very end, the dregs, is actually a very complex kind of cross-linked matrix of stuff that we actually don't really understand all that well. And that's due to sort of biological scavenging and sort of modifications of the particles as they sink in the ocean. The particle flux itself is shown on the right. There's some variability in the surface water that varies with productivity rates, but then eventually it all kind of decays down to very low amounts. And that's what we get as organic geochemists, is the stuff that's kind of gone through this massive filtering and transport process. This is a picture, not to scale, of the main conveyor of organics from the surface waters to the sediments. It's fecal matter, right? So all of this transport, the things I like to study, is like dominated by poo. It's not very elegant, but it's effective. So that's the marine side. And then I want to shift and then talk sort of in contrast the terrestrial side. And this is an image of where we find organic carbon. This is Rick Kyle and Larry Mayer's paper from 2014. It's a good review paper if you're interested in this. And they again have the marine realm sort of shown in these darker areas. That's where we have organic matter preserved, because there's a lot of production and sort of efficient preservation, perhaps from low oxygen conditions coupled with sort of high input rates. On land, we see also high accumulations of organic matter where it's cold. So wherever we can slow down decay, decay is destructive forces that eat up that organic matter and get it back into CO2 and leave me without a sample to work on, right? So we want to avoid the decay. And that is enhanced where it's cold, where decay processes are slowed down, and where you just have a lot of flux that sort of dominates and is wet and keeps soil from being well oxygenated. So in soils, we're looking for better preservation in cold or wet or both environments. You can think of the coal swamps, sort of iconic environments of terrestrial carbon preservation, because they're wet and highly productive, right? And in the peatlands and the tundra environments today, there's an enormous amount of soil carbon stored up because the decay of it is slowed by the cold temperatures, at least at the moment. In the marine realm, we have highly productive, as I mentioned, our environments where we have organic matter available for use. And then the anoxic environments are places where the lack of oxygen sort of limits or slows down the kind of decay that's possible. So these are really different contrasting environments where geochemists traditionally have looked for biomarker studies. And I'm a little unusual in my interests as an organic geochemist, because I keep going to the environments time and again in my career where these are not true. And so I tend to actually work in organic lean environments, which is really a sort of step away from the long tradition of organic geochemistry looking at these organic rich environments. There's a question.
Yeah. Would you also expect that in these cold, wet environments more recalcitrant carbon?
Recalcitrant, let me show you the next slide. Because she's asking me where would you find recalcitrant carbon? Recalcitrant is operationally defined as, carbon that's not easily reacted. And organic becomes recalcitrant maybe because of its inherent structure and properties as a molecule, or maybe it's stuck to a mineral surface that keeps enzymes from getting to it. So recalcitrants can be inherent or it can be environmental. And in the cold environments, it's actually not recalcitrant once you warm it up. It's actually quite delicious. And so there's actually fairly accelerated decay once it's into a realm where biological activity is sort of more active.
I'm just thinking in the global change, if they are subjected to more rises in temperature and you have less recalcitrant, then is that going to be more quickly released?
Correct.
Than in more stable ecosystems that have like intermediate environmental.
Correct. Yeah, no, this is actually, I think, an important point, and I'd be happy to talk to you more about this this afternoon. The change in environment can make what was once stable carbon unstable. And that's obviously a major concern as we're heating the high latitudes. There's a lot of carbon up there, and it's going to become unstable because the conditions have changed. What was once stable becomes less stable. In some of my, so some of my students who've taken this course, actually Shelby Lyons was here last year, she's working on deep time examples where there was warming. And we're finding that carbon was actually destabilized even in soil environments. So stability is conditional. And if you change the conditions, things that are stable can become less stable.
So you would expect that it depends more on the environment than on the liter quantity or?
Yes. Well, it depends on the boundary conditions, and if they change, then what we consider stable can become less stable. Yeah.
Thanks.
Yeah. And here, okay, so you anticipated my slide. This is from Schmidt's review paper, which is plotting mean residence time on the lower axis of different compound classes. And if you'll notice, all of these are kind of holding out in the realm of 0 to 50 years. So most organics and soils, are, as a geologist, I would consider not very stable, right? They're mostly reactive. But they tend to preserve on geologic timescales, probably for a variety of reasons, not the least of which is association with mineral phases. So they become less reactive when they're kind of rendered not as available to biology by interactions with the mineral phases. And so I study organics in ancient soils, and there's not very much, but what is there is not very reactive. And it's almost by definition. The reason it's still there is that it's not very reactive. And you see that orange, that bright orange color, it kind of complements your shirts. Those are some of the soil horizons we're really interested in, and they're really iron rich and they're very organic poor. And so we find ourselves sometimes measuring organic carbon contents that are not 1%, not 0.1%, but 0.02%, right? So we're really interested in these environments and trying to reconstruct them, and we're really straining our analytical capabilities with what's left behind. All of this is to say is that I study trash. So I've gone through this sort of exercise of talking about these different environments because I want to put into your head that when we study environments from the past through biomarkers, it's not a perfect recording of what's in the environment, but it's some kind of filter of process of transport and decay that leaves behind the things that survive those processes. And they're inherently less reactive. Often they're very big, so you can see the compounds on the left and on the far right-hand corner. Those have very low aqueous solubility because they're big molecules, and so their low solubility means they're less bioavailable for decaying microbes. Some of these others we find in different environments, they're of intermediate or maybe preserved because there's a very reducing conditions, for example. So I've spent my life sort of picking up trash and trying to characterize it. That's how it's not very glamorous, but really a life of great fun. And And it's important to be mindful of these, of what you're getting. And I think organic geochemists could take some lessons from the paleontologists who study fossils that are physical, fossils and macrofossils and even microfossils, because they've thought long and hard about this problem of transport and preservational bias. This is a really classic paper by Sue Kidwell and Flessa on taphonomy, and I've highlighted it and written it out in cleaner text because the paper is old and it's sort of a fuzzy diagram. But basically, we're moving from, the idea here is that what we study are the fossils that we sample, some maybe perfectly or maybe imperfectly, more likely the latter, and we're trying to reconstruct a living community and an environment. But to get to that place, the living community is an integration of time of many communities, conceivably, that then you have selected dead stuff that gets preserved and may get transported out, may get reworked, and then eventually gets buried in the geologic record and then is subject to further influences of diagenesis until it's what we would call a fossil, and then we can collect it. So I like this. It's not a perfect analogy for biomarkers, but it's an important sort of understanding to have as we think about what it is we're working with. It's the residue of a living community, and it's not one living community, but it's a time integration of many communities that we end up sampling. Okay, so what are our fossils? Well, our fossils are not bones like the dinosaur hunters, but they're the chemical constituents of cells, and we tend to get preserved those chemical constituents that are a little less reactive than others. And by a little less reactive, what I mean is that they're maybe not as nutritious or nutritionally desired by microorganisms. And the big one are the lipids. So cell lipids tend to, they're mostly dominated by hydrogen and carbon. They don't have, typically, at least not the polar head groups, aside from that, they don't have a lot of nitrogen in them. And so these things tend to be more refractory in the sense that they're less readily decayed in biological environments. So most of the biomarkers that we work with on long time scales are ultimately derived from some kind of lipid biochemical. So here's an example of that. This is on the top right or top left is a sterol with a hydrophobic portion here. That's the alcohol, and then this long kind of cyclic and branched hydrocarbon. And these are, of course, important in hormones and regulatory function, but they're also important in membranes, and they preserve pretty well without the hydroxyl group, and we over time lose those functional groups like the double bond. Hopane polyols are also very similar in sort of physical size, and they also seem to have some similar function, and they're found in bacteria, and there's related compounds in plants in ciliates. And so these have a biological form that then can be preserved geologically on deep timescales. Some of the biological forms are very diagnostic for their origin. Some of that chemical detail is lost with preservation. Here's some of the examples of phytosterols that come from plants and algae. And you can see these little ornamentations of the double bond and the hydroxyl group. That's often lost geologically, but we have things that might preserve, which is differences in the methyl versus an ethyl. There's another ethyl group there. We might or might not carry that double bond forward. So some of the subtle differences in the structures can actually be quite valuable in indicating a source, biological source.